I'm Leslie Wade. I'm program chair for the North American Fruit Explorers. Super excited that everyone is here. And I'm going to turn it over to Eric Bina, our facilitator.
Thank you, Leslie. Welcome to the North American Fruit Explorers 2022 virtual conference, Good Fruit from the Ground Up. Thank you for joining us. Today's session is Restructuring the Apple Family Tree, and I'm your facilitator, Eric Bina, a volunteer at NAFEX. And before we introduce today's panel, we'll quickly cover the basics that this is a webinar instead of a normal Zoom meeting, so your audio and video are often muted, but we do encourage you to ask questions using the question and answer tab at the bottom of your screen. And finally, this webinar is being recorded and will be made available to NAFEX members and conference participants after the conference at nafex.org. And for those who are new to NAFEX, we were founded in 1967 as a network of individuals throughout the United States and Canada devoted to the discovery, cultivation, and appreciation of superior fruits and nuts. While we include many professionals, we are all equal as amateurs and are motivated by our love of fruit. NAFEX members tend to share ideas, experiences, and fruit propagating material through our website or social media channels and special interest group meetings and annual conferences like this one. As a paid NAFEX member, you will get 4 editions of our Pomona Journal each year, as well as the ability to search our 50-year backlog of Pomonas in our digital library, which is an excellent source of information. Finally, this organization exists because of fruit-growing members like you, and we encourage you to continue your membership and become actively involved in an interest group or as a committee member or a board member. You can visit our website to learn more about this. All right, so let's introduce our speakers for today's panel. First, we have John Bunker, who's a very well-known orchardist and apple historian. He was the founder of the mail-order nursery Fedco Trees, and has devoted much of his time to searching for and preserving old apple varieties across the New England region. He's the author of the invaluable book, Apples and the Art of Detection, and founder of the Maine Heritage Orchard. And next we have Dr. Cameron Peace, a professor at Washington State University. His desire to match modern genomics and modern fruit breeding has led him to be the founder of the Washington Tree Fruit Genotyping Laboratory in Pullman, Washington, as well as co-director of the Rosebreed Project, which applies genomics to improved breeding in apples, blackberries, peaches, pears, strawberries, and cherries. He has many recent publications in DNA-informed breeding of fruit trees. And finally, we have Nicholas Howard, who is an apple breeder and geneticist at Fresh Forward Breeding in the Netherlands, where he's also a guest scientist at the Wageningen University and Research Center. He got his PhD while at the University of Minnesota in their apple breeding program and later did a dual postdoc there and at the Karl von Ossietzky University in Oldenburg, Germany. He is publishing and doing ongoing research in genetically characterizing the pedigree of modern and historic apples. So guys, tell us how you got together.
Okay, I'm going to go first. When I began my deep dive into apples about 50 years ago, it quickly became apparent that this was going to be about a lot more than the apples I was discovering on the old trees around town. First off, there were the names. As soon as I learned that there were more than Max and Red and Yellow Delicious, I knew that I wanted to learn them. And then as I began to get to know the names, I knew I wanted to know about the names. Where did they come from? Who were the people? And what were the stories behind the names? Fedco Trees started in 1984, and in about 1990, I did my first apple variety display at the Common Ground Fair in Maine. That first display was of roughly 50 cultivars that I just collected wherever I could. But it was a magnet that sucked people in. It was a big hit. So we kept doing it every year, and every year the display got bigger and bigger. And before long, people were bringing their apples with them to the fair in hopes that I would identify them. And so my love of apples forced me to learn to identify cultivars, as we say, phenotypically. and it simultaneously propelled me into history. The history of Maine, the history of New England, and the history of the world. Where did the apple come from? I read everything I could, and I began to piece together a narrative. I attempted to reconcile the contradictions of the various stories. Where it didn't jive, I thought long and hard, and then attempted to fill in the blanks, which were many. Over time, I developed a history which was pretty good in places and sorely lacking in others. Fast forward to 2018. I had met Thomas Chow, then director of the PGRU Plant Resource Genetic Unit, USDA Apple Collection in Geneva, New York in 2016. Thomas came to Maine for Maine Apple Camp in 2017, and we began to get to know each other. A year later, in 2018, I contacted Thomas about having an apple meeting in Geneva. He suggested tacking our meeting on to the annual Apple CGC Crop Germplasm Committee meeting, also happening that September in Geneva. A dozen or so Apple people from around the country would be convening there. Perhaps some of them would stick around for our meeting. Gail Volk, a research plant physiologist at the USDA Agricultural Research Service in Fort Collins, was one of them. Thomas, Gail, and I planned the meeting. It took place in late September 2018. There were about 25 of us. About half were apple historians, writers, and fruit explorers. The other half were scientists and academics with a major interest in apples. It was a fantastic meeting. One of the other attendees was Cameron Peace. Everyone in the room gave a short presentation. About 20 seconds into Cameron's talk, I knew I needed to get to know him. I'd heard about DNA profiling of apples, but really didn't know anything about it. At first, it terrified me. But once I began to learn about it, I realized that this could be the greatest adventure of my life, a collaboration between apple historian and apple geneticist. So far, it's turned out that I was correct. There are lots that I had figured out about the history of apples in Maine, and by extension, the US and around the world. But fairly quickly, I realized that the work that Cameron and Nick, who I've been beginning to get to know more recently, and others are doing, was going to be the tool that would be essential to fill in the gaps, sort out the contradictions, and correct the errors in assembling the apple family tree. Since 2018, I've been able to spend many hours with Cameron in person and via Zoom. A bunch of us from around the country put together the Historic Fruit Tree Working Group of North America, as well as the Registry of North America. We've been meeting twice a month as a group via Zoom. It's been a great collaboration. And so we come to today. We have, largely through the work of apple-obsessed historians and apple-obsessed scientists, the makings of an apple family tree. Cameron?
All right. So, okay, my turn to mention how I got into all of this and connect in with John there. So, well, I believe in fruit, and that's because fruit improves our quality of life, and it's an enjoyable way to ensure that people, and especially kids, receive the nutrition that we need. So that's why I went to university, to learn about and devote my career to fruit. My professional goal has always been to highlight and mobilize the inherent value of each fruit crop individual, from wild trees to cultivars to new seedlings. And while I enjoyed phenotyping fruit by eating them, I realized as an undergrad studying for a Bachelor of Horticultural Science, and that was at the University of Queensland in Australia, that a powerful emerging tool to address this goal was genotyping. And so that's DNA profiling and DNA testing. So DNA profiling being where we cover the whole genome all at once, and DNA testing is when we do it for specific genomic sites and the traits that they influence. So it occurred to me as an undergraduate that by genotyping, there was a compelling way that the value of any tree, and therefore groups of trees, such as all old cultivars or a wild population, could be highlighted. By highlighting this value, their conservation and usefulness would be emphasized and encouraged. This compelling way is to describe their utility in breeding, in contributing to future generations. That's future generations of fruit and future generations of people. So by doing it from this point of view, looking to the future, it amplifies the consideration of the value of any individual from what it is now and what it's been in past decades or even centuries to the many, many centuries of the future. So the natural step from there is to understand and appreciate the genetic connections, clonal identity, parentage, pedigree, and ancestry that unite every individual in a crop and a species. So it was with this ongoing goal of highlighting the value of each fruit crop individual that I became a foundational member and co-leader of the multi-million-dollar US-wide Rosebreed project, which ran from 2009 to 2019, as well as the US advisor to the Fruit Proteomics project, 2010 to 2014. That was by our colleagues across the pond in Europe. So these projects developed, extended, and applied the powerful genotyping tools and genetic value uncovering approaches that we now routinely use. And I met Nick when he came to one of our annual Rosebreed meetings in February 2017. And of course, he'll tell you shortly about what he'd been doing up to that point and where he's gone from there. And then in September 2018, that's when I met John, as he mentioned. And I offered the opportunity at that point for DNA profiling to John and to Laura Seeger and Todd Little-Sebold of the Maine Heritage Orchard, so they could, and all of us together collaboratively, we could check out some of the interesting things in their collection. And by then we had some interesting stories that I was enjoying telling about the parentage of Wealthy, for example, and Granny Smith, Red Delicious, some of these old cultivars we'd already been uncovering their parentages. And I proposed to them that apple cultivars were all related within a handful of generations to pique their interest. So then as Rosebreed ended and each breeding program went their own way with what was learned, in my research program, we expanded the application of the DNA profiling technology for apple and cherry from those large breeding programs to anyone with trees of value. So that's of value to you now and of potential value to future generations. And so for the past few years, we've been receiving and processing leaf samples from apple trees in collections, old homesteads, remnants of abandoned orchards, backyards, sides of the road and tracks, national parks and forests, all trying to figure out each tree's identity, parentage, and pedigree position. And you can see for more information, www.myfruittree.org. And we do this as a public research endeavor. It's not for profit. We do this on the side while in my program here, while we pursue our other responsibilities with our research projects, teaching, and professional service. And the genotyping by myfruittree.org, it's a component of the approach by the Historic Fruit Tree Working Group of North America that John was mentioning, where we meet every couple of weeks for the last several years. And the mission is to facilitate the conservation of historic fruit tree cultivars in North America through documentation, identification, collaboration, and education. And with that, I'll end my introduction.
Yes, as Cameron said, I met him at the Rosebreed in 2017. Before then, when I was a undergraduate, I decided real early I wanted to get into plant breeding, and I love fruit. I wanted to specifically try to get into fruit breeding. And so when it came time to do my PhD, I'm from Wisconsin, so I studied at the University of Wisconsin. And when it came time to do the PhD, I contacted Jim Luby at the University of Minnesota. Actually, originally to work with grapes. I was interested in grapes, but he had an opportunity to work with apples. And I said, hey, this is the guy that develops the Honeycrisp apple. Sounds like it'll be a good experience. And I quickly got onto the idea of what's called pedigree-based analysis from a sort of championed by a scientist here in Wageningen, where I'm at right now in the Netherlands. And so I learned about this through the Rosebreed meetings. and working with people like Cameron. And I got involved in this project to identify the true pedigree of the Honeycrisp apple. And so Cameron and I think Jim Luby had known or had an inkling that the grandparents were through the unknown parent were Golden Delicious and Duchess of Oldenburg. And I went through the genetic information and the records and things at Minnesota and was able to identify what the true parent was. It doesn't exist anymore. It's an old breeding selection. But then after making this discovery and then applying it to my other work, which was involved with looking at the correlation between areas of the genome and traits, I found that this pedigree reconstruction work was very useful for breeding. And it was so useful, in fact, that I thought this would be a great topic for my postdoc. And specifically, I wanted to see if I could identify how North American cultivars are related to their European progenitor cultivars. And in order to do that work correctly, I felt I needed to move to Europe to do a postdoc. So there's opportunity in Oldenburg, Germany. Moved there in 2017, 3-year postdoc there. And I just started asking everybody throughout the world that had genetic data on apples, specifically SNP array data, asking them if I could have access to it, and started pooling it all together and seeing how it's all related. And the original goal of finding the European ancestors of American cultivars ended up just growing to the point where I just organized a giant database of apples and started organizing the duplicates and how everything's related to one another, and developing with some collaborators techniques on how to process the data and everything. And it's been this loosely defined, loosely organized, worldwide collaborative effort to figure out how everything's related to one another. And then through my interaction with Cameron, is how I've relatively recently met John Bunker. And then more recently, the past couple of years, I took a position as apple breeder at a company that's partly connected to the University of Wageningen. Or Wageningen University and Research is their full name. Yeah, so that's it for my introduction.
Okay, Cameron, you had some slides you wanted to show to get us started?
Yeah. So, let me get a moment. Do one click. There we go. You can all see that, right?
Yeah, it's good.
Okay. All right. So, yeah. We're going to talk to you now about this topic specifically. So not just reconstructing the apple family tree in general, but particularly of US apple cultivars is what we want to focus in on. And so the idea is for this to be informal. So the three of us, that's John and Nick and I, are going to chat with each other and throw out facts and hopefully, debate, and we'll see where it goes, but also take questions from the audience all the way through. In fact, even right now, just based on our introductions and anything, I think is a good time already to take any questions if anyone has any. Start off.
Yeah, it doesn't look like you generated any questions in the Q&A on the intro. I think everyone's been too busy listening to ask questions.
Yep. Okay. Well, you got your prompt now, everyone. So yeah, think of what you want to ask, and Eric as well will introduce us. So interrupt us at any point here too, if something seems appropriate for us to answer in the thick of it here. So the idea is, again, for it to be informal. So, origins of US apple cultivars, discoveries and speculations from DNA profiling. We've only got a couple of slides here. I mean, basically, we're just going to talk. It's not really about slide. But we will assume that you either are at least vaguely familiar with the resources, the three things that are indicated here, or you will be soon after. And so a lot of the things that we talk about will make sense if you check out these soon after. I recommend you watch them before. So if you're watching this via a recorded video, I'd say stop now and go check out these other videos first and check out the paper here, and then come back. Things will make a lot more sense, I think. So here provides, and we'll probably come back to this particular slide several times, provides the 6 premises that what we're going to be discussing is based on. And we won't necessarily be hitting these up in this particular order, but roughly we will. And so we're going to be explaining and describing again amongst ourselves, but how everything pretty much fits into one big family tree, and that the vast majority of American cultivars are connected into a single pedigree, even that's via in-laws in many cases, especially as they get older and older. That there's only a few founders that we know, but still things are connected up. There's a lot of common individuals, and in fact, I'll just go to the next. No, I'll come back to that one. But there's a few key known and unknown founders. One particular, the most important one, is this Rainette Franch, grandmother of all. Talk about that a fair bit. And then there's a few generations of unknown intermediates after Rainette Franch in particular, with some missing individuals in there. And that's, for me, I think that's a really exciting part to try and figure out what's going on there. There's still a lot of unknown ancestry, so there's lots of material that comes in to the pedigree that is unaccounted for, and you can help as well. So this is not just us telling you things, but you can contribute to this also. Any comments from John or Nick at this point? The next thing is just to give a brief explanation of what the SNP arrays that we use, the DNA profiling tool.
You did have a question, which you may be answering soon anyways, which is how diverse is the genetic background of U.S. apples? How many progenitors are the base of most of U.S. cultivars? Yeah, all right. So that's what we're going to get into. So I won't give a quick flippant answer. We'll be getting all into that. That's one of the big ones that we'll be covering here. Great question. Mm-hmm. Hopefully you're satisfied by the end of this about that. Okay, so this is a slide here from one of those talks, just explaining quickly the DNA profiling technology that we use that Nick had mentioned just before, these SNP arrays. And so we do the DNA profiling with these, and it's a set system of running these genetic markers, of the same genetic markers all the way down the chromosomes of any individual that we run. And along each chromosome, there's 17 chromosomes of apple. Along each one, there's hundreds of these SNPs that we use, or that are targeted, that we get the information for. And so with each one, that reveals any genetic differences that there are, and that's, as it says there, between the two chromosomes that an individual has, two pairs of each chromosome, and it got each member of those pairs from its mother or its father, one from the mum, one from the dad, and so that already provides some differences. And then comparing that to all the other individuals that this is run for. And because we have such a high density of these, it makes it really powerful. And so this superseded a previous technique, which was SSR panels that were used. They're still used for simple situations, but it's really powerful what we have. We can go well beyond just identity, well beyond just saying, is this the same as that? And so that's where a lot of the insights that we're going to be talking about come from, is the ability to go up to parents and then grandparents and other things like this, and skip generations. So I'm going to interrupt with a question because I think some people may also be interested or confused by this, since they're calling these SNPs, which, talks about cutting. So Pat Hallen wants to know if you guys breed, for all the panelists, do you do your breeding by genetic engineering with CRISPR, or do you use old-fashioned breeding techniques of crossing two parents?
Okay, so the SNPs part is just the way it's pronounced in this. And also this has got nothing to do with breeding, what we're showing here. This is just diagnostics. This is just looking at stuff. So anyone, could do this if you just run the genotyping tools, if you had access to it. So people who are not even doing breeding can find out about their material in this case. But specifically about those folks who are doing breeding, such as Nick, but also the University of Minnesota and Washington State University and Cornell University, the three big public breeding programs of apple in this country. It's not genetic engineering, it's just traditional crossing and then selecting among the kids. And the genotyping that we do is just a diagnostic, easy way. It makes it more efficient to identify the genetic potential of any given individual.
Great. And someone wanted to know the definition of SNPs. Like, what does SNP stand for? Single
nucleotide polymorphisms. So the nucleotide is just an A, C, G, or T part of the sequence along the chromosome. And so when you have differences, that's the variation that we have between them. So one individual might have an A and a C.
Great. And then one final question. Mark Kruzmak wanted to know if you take your SNPs from a single chromosome, which looks like in this picture, whether you're taking them from all 17 chromosomes.
Yeah, so all 17, and it's, we're not taking anything, it's just looking at it. Like the picture that I had back at the beginning here with the magnifying glass, looking in at the DNA on the leaf there.
Great,
so that's
thanks.
that's effectively what we're doing, just looking in at each particular one. Yeah, all along each chromosome, well, the two pairs of each chromosome, as it's showing here for chromosome 1, has got two pairs from mum and dad, and then for the other 16 chromosomes as well. Nick might well want to say some things here too, especially regarding the breedings, given that he's actually a breeder, an apple breeder.
Yeah, sure. So the kind of breeding I'm doing is traditional, and that we're not using any sort of genetic modification techniques, but this genetic information is extremely useful for developing varieties in modern breeding. So, like, let's say I want to make a really disease-resistant variety. A lot of the resistance breeding for scab resistance, scab being in many places the most important fungal disease of apple. If you want to combine different major gene sources of resistance, it's the only way to effectively do that is to use genetic markers. So you go and profile your individual and see which resistances are there. There's other ways to do it without markers, but this is the most effective, fast, easy way to do it, the most efficient. And in the sort of breeding I'm doing, we're targeting lots of different traits, and getting all this SNP information helps us understand what sort of traits, what Cameron said, the genetic potential of an individual. It's really, really useful. And just a little more context information to add to this slide. We're in the data we're working with right now, we're looking at 10,000. It's 10,321 SNPs currently in the present database. And just to compare that to how people are doing similar things with humans, if any of you are familiar with the commercial genotyping services for humans that use SNP arrays, they're using on the order of hundreds of thousands. If I recall, the last version of the array has several 100,000 SNPs for humans. Yeah, so we're using 10,000 for what we're doing right now.
Okay.
And I see somebody saying I suggest defining the nucleotide. Yeah, the nucleotide is just DNA is a sequence of A's, T's, G's, and C's. So A SNP is like there's a sequence, and then one part of that sequence differs at one of the nucleotides. And so a SNP array is an array of probes that target different SNPs. So they have that sequence right next to the SNP. And then it checks whether it has the A variant or the G variant, for instance. And that for tens of thousands or thousands or tens of thousands, whatever SNPs.
Yep. Okay, so yeah, some of those questions might come up in, or might be relevant for, come up in more detail for some of the things we're going to say now. So again, here's a slide that was from talk I gave a couple of years ago, but we'll use this as a launching point for some of the stuff that I think Nick's now going to tell us about, and especially focusing in on, well, it should say Rainet Franch is the real name of it. That was, so when I gave this talk, I was just pointing out that there are some key founders that have come up, as it says, some discovered or imputed. So Rainet Franch is the parent, and then the grandparent and the great-grandparent of so many things. We'll talk about that in a moment. And then some others that Nick, from gathering all the information, lots and lots of DNA profiles, was able to impute, which means, well, he can mention what that means, but it's not necessarily an actual individual that we know. It's prior to even having an individual exist, a named cultivar, but realizing that it's the parent of a whole bunch of things. So I think might be time for us to talk about Rainet Franch, and then also John can get in at that point and talk about how this could have come about, that Rainet Franch is the parent and grandparent, the founder of so many things in this country.
One thing that I could say first before we get into Rainet Franch is that one of the questions that came up early was about how many, what is the origin, what are the origin varieties and so forth, and one of the things that Nick and Cameron and I were discussing the last week or so when we were thinking about this talk is that the apple came to North America from different sites in Europe, and so I was talking about, and I'll tell you a little bit about how Maine, where I live, is sort of a microcosm of the U.S. And we had here three major sort of arrivals, we'll say, and groups. One of the arrivals, which is the one that we're going to talk about first, I think, of Rainet Franch, those were the apples that came with the immigrants primarily from England, the Mayflower and all that. If you think about the Mayflower and the Arabella and the founding of Boston and all that, that was all. Those are the classic New England varieties. Then we have another group of apples that came over mostly through Canada, and those apples, which are somewhat different, include Macintosh, Famous, St. Lawrence, and other varieties that either originated in Canada or came from Canada, and then in the case of Maine, came south down through northern Vermont, New Hampshire, and Maine into southern New England. That's another whole group. And then the other real major group that came to Maine, which also came to many parts of the U.S., are the Russians, and or the four classic Russian or Russian-related cultivars from the 1830s, and those would be Duchess or Duchess of Oldenburg, Alexander, Red Astrakhan, and Tetovsky. And then later, a bunch more came in around 1870. Those included yellow transparent, in particular, one of the other famous ones of those. So we have these three different sort of groups of immigrants that came in, and that sort of maybe sets the stage for us to look at Renette Franche.
So, Nick can go into more detail here, but what we basically have from Renette Franche, which is a 500-year-old cultivar, as it says there, or at least it was first described, in 1510, that there are three sort of levels of descendancy from this particular cultivar. First is few rare individuals, American cultivars, that are children of Renette Franche. Then there's a bunch more, a dozen or more, that are grandchildren, and that is where we don't know the intervening parent, the intermediate ancestor there, something that doesn't seem to exist anymore, or we haven't discovered it yet. There's a whole bunch that are grandchildren, many of which are cultivars that you'll recognize the name of. And then there's the next level, which is beyond grandchildren. So that's where Renette Franche is probably a great-grandparent or a great-great, etc. And again, with missing ancestors in between. Of course, there's plenty of descendants then from the ones that are the children, many descendants from the ones that are the grandchildren, all of that. But there are three levels there. And so the individuals that are the children are quite interesting because they help us figure out, give us big clues about what might have been going on for how this happened. And then same with the grandchildren. We try to date those when and where they popped up. So Nick, can you tell us more about let's start with the children of Renette Franche?
Sure. So this first, the identification of Renette Franche as an important founder of American material isn't too surprising because it was previously identified to be an important founder of lots of European material by a group at INRA in France. So there's a paper published in 2019, Moranti et al. And yeah, so they found that Renette Franche is a parent of or a grandparent of many cultivars across Europe. And in their study, they included the Esopus Spitzenburg, which is a very old American cultivar, and that is one of the direct offspring of the Renette Franche. And more recently, I published with some collaborators a paper on pedigree reconstruction of triploid cultivars. So these are cultivars that have 3 copies of each chromosome as opposed to the normal 2 copies. And the oldest U.S. cultivar is recorded as being Roxbury russet, a triploid. And Roxbury russet is a offspring of the Renette Franche. And we're quite sure that the mother must be the Renette Franche, as I described in the paper. You can kind of know with triploid apples and the genetics. We generally believe that the mother of triploids donates 2 copies of the chromosomes, whereas the father donates 1, and that's the case here with the Roxbury russet. And so those are 2 very old cultivars with Renette Franche as one parent, coming from presumably the earliest European settlers into New England, bringing apples.
Yeah, maybe I was going to say that maybe before we go beyond that, maybe John, if you could give us your speculations about the origins then and that connection of Renette Franche with Roxbury russet and also Esopus Spitzenburg.
Well, one of my favorite apple little bits that I love to read, and when I give talks, is the one of Jim Hawkins on the Hispaniola or whatever the name of that ship was, when they come over for Treasure Island and he's hiding in the apple barrel. And then here's the plans of the pirates. And that apple barrel was on every ship. And you have so that got me thinking, okay, so when the ships left England, they mostly left at the end of March, and I think that had something to do with the winds, and something even more to do, of course, with getting people to Massachusetts by June. And an apple, Reinette Franche, is a very late keeping apple. And so when they provisioned the ships that came over, and now we could think about John Winthrop, who came he left England in early April 1630 with seven ships, or 11 ships and 700 people, and they were really the ones that founded Boston, Roxbury, and Dorchester. They would have left right when Reinette Franche and other late keeping apples would have been at their prime. So when they put the barrels, when they provisioned the ships, the Arabella and others, with their food stuff, they would have put barrels of presumably Reinette Franche right on deck for the passengers and the crew to eat. So they ate the apples as they went across the Atlantic, and they kept the seeds in their pockets, or some of them did. And then when they got to Salem and then Boston, they would have, theoretically, and I think probably someone, and it's still a bit unclear as to who that was. There's a bunch of names kicked around Roxbury Russet, but somebody would have saved the seed, planted the seed in the spring of 1631, and then the apples, they would have sprouted that spring. And apples from seed, as those of you know who have grown apples from seed, take about, we'll say, 9 to 14 years to produce their first crop, or their first decent crop. That would have taken us up to about 1645, and that's about the time that the existing histories of Roxbury Russet say that it originated. So that all sort of fits with my sort of speculation, we'll say, and I'm still working on it and still reading what I can. But my theory is that it was Reinette Franche as apples in barrels on the ships coming over. The seeds were saved, planted, and then that gave us Roxbury Russet. As far as Aesopus Spitzenberg, as far as we can tell, Reinette Franche really wasn't grown from scionwood, from clones in New England. So I haven't tried to follow the Aesopus story in detail yet, but it must be somewhat similar. The fact that Reinette Franche doesn't show up as, in many cases as the parent, but does show up as the grandparent or great-grandparent of many American cultivars would suggest that what we know is that most New Englanders planted their apples from seed. And so we also know, and Nick was talking about this the other day, which I'd like to hear him say more about too, perhaps, is that many of those first seedlings from England, maybe even from the mainland too, did not do well in North America. And that meant, however, some did. So we have this, and if you've read your Henry David Thoreau Wild Apples, we have this culture of hundreds of thousands of seedlings all over New England. And then the best of them would have produced children who would have produced children. And then you start to get the selections of New England apples, that many of which wind up with Reinette Franche in a little more distant ancestry. So there's a few thoughts about that.
So I'm going to interrupt quickly with a historical question someone asked that this segues well into. Al wants to know what apples from the Basque area might have been brought over with the fishing fleet in the early periods, and would Basque or Spanish apples show up with different genetic markers?
So I do have some Spanish apples in my database, and there's been a number of papers published on the diversity of Spanish material. Well, in general, Iberian apples are genetically very distinct. They're in general very different from other areas of Europe, though they do have some of them are derived from the Reinette Franche as well, or partially derived from the Reinette Franche. But as far as I can tell right now, I've not identified any direct links between Spanish apples and American apples, though the number of Spanish apples in the database currently is rather small. I'm working on increasing that by reaching out to some other scientists in Spain, however. But they're so different genetically. I think that if there would have been some connection, I would have been able to identify that with the techniques that we're using. That's not to say that there aren't any connections, and there might be some more connections with southern apples, but there's also southern U.S. apples, but there's also a lack of southern U.S. apples in the database as well. So for right now, no, I've not identified any. There might be some connections though.
One of the things that we should do in the coming years is go to Red Bay up in Canada, where the Basques were fishing for whales and cod, and find remote seedlings and then do DNA profiles on them, because one possible location for where there would be Basque material or material of Basque ancestry is in that area of maritime Canada.
So I think talking just a little bit more about the Reinette Franche will then bring us to the other founders we want to discuss. So as Cameron was saying, some of the early material that came over was direct offspring of the Reinette Franche, but a lot of the material that was brought over, likely as seeds, as John mentioned, would be planted, and then the climate in what became the United States is quite a bit different than a lot of areas in Europe. And so America became another area of genetic diversity in apples because lots of material was brought over, and there was a lot of seedling experimentation to be able to, as I understand it, grow cider. And apples are also a cheap source of sugar for baking and all this and that. And so the settlers would want to have apples around. They plant the seeds, the material that was able to withstand the climate and was good enough to keep around, they kept. And then that begat more material. And so some of the first generation descendants of the first material that was brought over as offspring of Reinette Franche then would be the grandchildren of Reinette Franche. And I saw one of the first questions here was about the Black Oxford origin. And the Black Oxford, one of its parents is let me just check quick, is the...
Yeah, I just sent that. It's blue pear Maine and
Blue
Hunt
pear
Russet.
Maine. Yeah, and blue pear Maine is a grandchild of the Rhinette Franche, as is a really old cultivar I'm not familiar with called Eustace. I think the two Blue Pear Maine and Eustace are half siblings sharing Rhinette Franche as a common grandparent, or as a grandparent through a common parent. And so as apples were proliferated and spread about, we have then fragmented Rhinette Franche ancestry through all this material. And so later material coming from the 1800s or so, we can tell that Rhinette Franche is an ancestor, but we oftentimes don't know the exact relationship unless we have some of the intermediary generations. Jonathan, for example, of course, is an offspring of the Sopa Spitzenberg. And so then we can trace that to the specific relationship. But some material like Rome Beauty, Ben Davis, material like that, we're not quite sure what the exact relationship is, but we know that there is a relationship there, several generations removed. Another founder related to the Rhinette Franche that is sort of a similar story is the Rhinette de Carme, or as it's known in Germany as Carmelita Rhinette. And there's a number of direct offspring of this individual. Cameron mentioned one that was the Hunteresset, right?
Yep.
Yeah. And the Rhinette de Carme is in the pedigree of Jonathan, but it's another situation where we don't know the exact relationship. It could be a grandparent, it's more likely like a great grandparent, but it makes it a little difficult because there's some shared ancestry between Rhinette Franche and the Rhinette de Carme, and we don't know the exact relationship between them there, but they share a significant portion of their SNP data.
In that case, Nick, what might it be? Like as close as a half sib, or maybe or is it like one generation further than that?
It'd probably be one generation at least further than that, possibly two. But they're both French apples, I think, originating in a somewhat similar time frame. No, Rhinette de Carme originated a little bit later, if I recall. But once you get that old, who's to tell how old the material really is? Because a lot of times the records are like, this was first recorded in X year. But that's maybe the first time somebody bothered to write it down.
Yes, and in those cases, sometimes the cultivar has been around for many decades at that point. One of the things that's really interesting to me about Blue Pear Maine is that for those of you who are interested in early American apple history, Blue Pear Maine is one of the ones that comes up as being just a big question mark. And that always suggested to me that, one, it's very old, and two, that if it did have genetics that were closely, related to the European apples, it would be a good candidate. And the fact that if you read about Blue Pear Maine's history, it's mostly speculation until now. It totally makes sense that it would be a grandchild of Rhinette Franche. And that probably means that it originated, maybe even before 1700, but if not before 1700, early in the 18th century. I could also just say that in Maine, when we find ancient grafted trees, the oldest trees, one of the ones we find typically is Blue Pear Maine. And the other most common of the ancient varieties in Maine is Tolman's Sweet, which maybe these guys will talk a little bit about Tolman's Sweet.
Go ahead, Cameron.
Yeah, Nick, go for it, but I'll just ask Eric a question on the side, which is just if there are any more, and I know there are some more appropriate questions from about Rainet Franch, and then otherwise, yeah, Nick will get into the next other founders.
Sure. So my for each apple apple whose SNP data I receive, I try to identify what it's related to. And so my one note for Tolman Sweet is that Rainet Franch is at least one grandparent. So that means that it might be in the pedigree more than once. And the grandparent-grandchild relationship would need to be confirmed. It could be that Tolman Sweet Rainet Franch is in the pedigree of Tolman Sweet numerous times, and so it just is really, really related to it. But it's likely a grandparent. We can confirm this if we have enough offspring of Tolman Sweet to do what's called marker phasing and then analyze it more closely. But it also is likely a close descendant of the Rainet Franch.
Okay, one more Rainet Franch question, which is basically someone asking for clarification of was Rainet Franch itself ever brought over as a scion, or has it only been here as offspring from seeds, as John described?
I don't think that early on it was brought over as scion wood. Maybe it was. I don't know how exactly the seed savers got their sample. It's also in the Geneva collection. Maybe they have notes on how it was brought over. If it was sampled but from somebody in the States, or if it was brought over from somewhere in France or the UK or something, I don't know. But I'd imagine early on, like what John said, that the materials brought over as seeds.
And then Chris O'Mannix wants to know if you know any of the parents of Rainet Franch itself.
Yeah, so Eric and I were actually talking about this before this session started. There, in that Moranti et al. 2019 paper, they posed one individual as potentially being a parent, based on I think it was based on the literature, but through further analysis with new tests, we can confirm that it is just another offspring. So we haven't identified the parents of the Rainet Franch, but I have identified a group of half siblings that includes Rainet Franch. And so when you have marker data for numerous individuals that share an unknown parent, you can identify them as a half sib group using genetic techniques. You can do what's called marker imputation. And so I have a genotypic profile for this unknown individual, but we've attempted to identify what it is through working with some of our collaborators in France, but we've not identified what the individual is right now. At one point, there was a parent of Rainet Franch that also was grown widely and begat numerous extant offspring that have been genotyped.
Okay.
And Rainet Franch too, I saw a question. It is a pretty good apple. So it's not just happenstance, like it just happens to be there. It's a pretty good apple. I think it was kept around for its. I think John said that it's a keeper. It's also quite juicy. It's got a good flavor. It's nice balance. I rather like it. I've had it a couple years here while living in Europe.
Okay, and more questions just keep coming in about Rainet Franch. But Todd Little-Siebold says that Rainet Franch was brought over in the 1830s by Robert Manning from France. So I assume he means scion was brought.
Yeah, and then for it being the parent of Soper Spitzenberg and Roxbury Russet and stuff like that, one of the as Nick was saying, there isn't evidence, or we'd imagine or think that reason that it only came over as fruit, as seeds, rather than the sign itself way back prior to 1830, because what we can do is consider the other parent, right, like a Soper Spitzenberg and the other parent of Roxbury Russet, because if whatever that is, when that gets extracted out, we look at that DNA profile. If Rainet Franch had been brought as sign wood, as a grafted tree to this continent, then the other parent of a Soper Spitzenberg, for example, would be something that was here as well. But in both those cases, those two cultivars, Roxbury Russet and Rainet Franch, the imputed other parent is not. It's back from Europe. So far, no evidence that the rennet Franche contribution was directly from the tree being grown here.
Okay. And then
I
Chris,
see one quick question about the half sibs of rennet Franche. They're all, at least to me, obscure cultivars from France and Italy. And so probably you wouldn't recognize the names. Tarella, Franc, Rousseau, Michelet, Pupino, Ferreira. I don't speak Italian very well, so yeah, they're really obscure sort of cultivars. They're old. I looked briefly into them. A lot of them are quite old. There's 6 or 8 of them.
When I started to learn about rennet Franche, it totally made sense that it was not just a random apple that they were collecting seeds from, that they would have selected it because the fact that it is a top quality storage apple, and that storage apples are just so valuable when you lack refrigeration. And Roxbury Russet, along with, really, we could say Baldwin and Rhode Island Greening, but Roxbury Russet was extremely important in American apple growing for over 200 years because of its ability to keep well in cold storage for many months. So when I was so surprised when I heard from Cameron, because I hadn't met Nick yet about this rennet Franche, and I'm thinking, what is this apple? I've never even heard of it. And then I thought, well, okay, so what was so great about it? And it totally makes sense.
Okay, so so many great questions here. There's no way we can get to all of this. This is the sort of stuff that why John and Todd and I and others meet and the historical fruit tree working group of North America meet so regularly, because we just talk about all these things all the time. There's just so much interesting stuff here. So sorry for not getting to your answers by the end of this thing, the end of this session, and you can email us afterwards. Would be a useful way for some of your key questions there. Maybe we move now into the other founders.
Okay, yeah, we should. One thing that we should do, because of how important they are, is we should at least talk a little bit about McIntosh, Famous, and St. Lawrence, and we should also talk about Duchess of Oldenburg and Alexander, and Wealthy and the Midwest, and so those other immigrants from Europe.
Yeah, and unknown founder 13. So yeah, that's your cue, Nick.
Yeah, sure. So let's start with let me pull up my notes here. So let's talk a bit about the other major European founder that hasn't been mentioned is the Calville Rouge. And so Calville Rouge is a French cultivar, really wonky shape, very, very red. Also identified in Moranti et al. As an important founder of European material. It isn't, I don't think I've identified it as a parent of any American cultivars, but it is sometimes a grandparent, and it is sometimes otherwise in the pedigree of, in the genetic background of American material. So a couple examples are Famous, which is a Canadian cultivar. It's if I recall right, it's a grandparent of Famous. Famous we showed in a paper that Cameron and I and some collaborators wrote or published, I think this year or last year, I can't remember, that that the Famous is one grandparent of Macintosh. Macintosh also has this Calvillo Rouge coming in it through into its genetic background through some other source as well. And Jersey Black, I think that's the name of the cultivar, is also a grandchild of the Calvillo Rouge. And so John kind of mentioned this material coming up from Canada that into what's now the United States, and I think that material has a bit more of the Calvillo Rouge sort of background. The St. Lawrence, right? Well, that was St. Lawrence itself is it's an offspring of Famous. So yeah, that material is going a bit from Calvillo Rouge. Otherwise, beyond that, the extant founders, John mentioned the 4 Russian cultivars that were brought over in the 1830s: Duchess of Oldenburg, Kaiser Alexander, Tatovsky, and Red Astrakhan. And Red Astrakhan, of those 4, is sort of the odd one out. It has a little bit of its ancestry coming from that Calvillo Rouge, but it has more of a Scandinavian genetic background. It also is not featured so prominently in the pedigrees of American cultivars. A few, but not quite so much. More important were the other 3, specifically Duchess of Oldenburg. And this year, we published a paper. I published a paper with my folks I worked with previously at the University of Minnesota on the extended pedigree of the University of Minnesota cultivars. And Duchess of Oldenburg was identified as in the pedigree of every single one of them, except for one obscure cultivar release. Every single one except for one. Tatovsky was in a number of the pedigrees as well. And Tatovsky and Duchess of Oldenburg are very closely related, possibly half siblings. Kaiser Alexander is famous for making really, really large apples, and it's in the pedigrees of a number of individuals. Trying to remember Wolf River, right? Wolf River. It's one of the parents of Wolf River, for instance, which is probably why Wolf River is a giant apple. It's getting it from Alexander.
Yeah.
And Tatovsky is Beacon, for instance, is a grandchild of Tatovsky. Tatovsky was used a lot in breeding in Canada. So if anybody's familiar with Melba, it is I think it's a parent of Melba. Yeah. And it's also in what's that cultivar called? Monarch. No, not Monarch. I can't think of the name right now. It's
Burgundy. You're talking about what Melba's in the background of?
No, it's Tatovsky is in a cult in the back it's a parent of
Oriole, was it?
Not Oriole. Name has escaped me right now.
Mantet.
There we go. Mantet. I don't know how widely that's grown in the States, but it seems a lot of folks grow it around here in northern Germany and in the Netherlands. It's extremely early. It's one of the parents of that. Yeah, otherwise, other extant cultivars. I identified a couple links with Oppie, which is known in the States as Lady. So this is recorded as an ancient French cultivar, and it's very genetically distinct. Its genetics are more Italian, and it might be a grandchild of a Roman cultivar that we're working on properly identifying. So Oppie is one of the grandparents of Famous, and often the genetics of Oppie and Calvillo Rouge were coming together into the American material. I don't know why that is, just by happenstance. Oppie was grown a lot in France, as is Calvillo Rouge, so they crossed together and seeds were brought over, something like this. Other than that, for known direct relationships we can identify, those are the founders where we've identified, but besides an unknown what we call unknown founder 13, we're working on the identity of that as well. But there's a few instances of obscure connections. So lots of immigrants came over to the United States, and some of them brought seeds of their favorite fruits. And so there's a few connections with, for example, German cultivars. And a couple examples here are, there's an American cultivar called Hahnemann. Hahnemann, I don't know if I'm pronouncing it right. It's an offspring of Prinzenapfel, which is a very weird shaped, like almost cylindrical apple that is a common founder of German material. Richland crab is an offspring of Zigeunerin, which is an apple I can get in the spring here in the Netherlands at like specialty grocery stores and things, organic shops. And the cultivar Garden Royal is an offspring of Schleswig Erdbeerapfel. And I cannot pronounce that. It's Schleswig, the area between Denmark and Lower Germany. That's what the Schleswig is. So the apple presumably comes from that area. And there's a few connections with crab material. So there's this cultivar called Manchurian crab that was brought over. This is genetically very, very different, very, very winter hardy. So at some point there was some interest in crab material being brought to the Midwest for use for breeding because of how harsh the winters are. Manchurian crab is one of these, and so there's a few connections there. That's it. That's it for the extant connections. Unless I'm forgetting any, I think that's it.
And I think we could just say to those of you who aren't familiar with Duchess and Alexander and Tatovsky and Red Astrakhan, that was all organized by the London Horticultural Society and the Massachusetts Horticultural Society to get cultivars over to North America for those who were moving to areas where the most apple cultivars could not be grown because they just weren't hardy enough. So it was a strategic move to get hardier material into North America, and it worked.
Yeah, through some of the things that Nick was talking about, I should have had this up. This would have helped some of that. We've only got this, and then the next slide with some other things to help out. So let me just go to this next one here, just so maybe Nick can point out in terms of what he was just talking about or what this is showing. And it's Macintosh.
Yeah, this is from one of our recent papers, and the highlighted colored portions show areas of the genome that are in common between Famuse and Macintosh. Specifically, it shows kind of all the chromosomes of Macintosh in the highlighted areas are those that are identical to the same areas in Famuse. And areas where it switches from one color to the other, where they're extended, that can be areas that show evidence of a grandparent-grandchild relationship. That's a recombination, as they say in genetics. So that's where you have, I don't know how much we want to get into the genetics of it, but
no, we don't.
Yeah, we don't get too deep into it. But that it's really good evidence, have like on chromosome 1 and on chromosome 15. That's really clear evidence that we have a grandparent-grandchild relationship. The amount of coverage and having those recombination is great evidence for grandparent-grandchild relationships. And so when we see things are closely related based on the genetic information, we have to kind of zoom in on these sort of things to clarify the relationship. Sometimes it's unclear, but in this case, it's pretty clear. So maybe you want to go back to the other slide here. This is a bit of a mess of a figure. This is from the papers shown in the bottom right. If you want to go check it out, it's open access. And this shows a few of the relationships we were describing. So Rennet Franche is a grandparent of the yellow bellflower. Yellow bellflower is a grandparent of Golden Delicious. The known parent of Golden Delicious is Grimes Golden, and Rennet Franche is also a grandparent of that. So Golden Delicious is a little bit inbred. And the other line going from Rennet Franche is to Northern Spy. That's the only connection I found with Northern Spy. I've not identified either of its parents. Or we've not identified any of its parents.
So Nick, I have to ask, when you did that yellow bellflower chain, do you have techniques that show that it's definitely only one generation between Rennet Franche and yellow bellflower, and again, only one generation between yellow bellflower and Golden Delicious? Or could there be, several inbred children that then cross in there to get the same amount of genetics in the apple underneath?
Sure. So first, in apples, we don't really self. I've not identified any genotypic profiles that show evidence of selfing, except for 2 samples that were intentionally created in a greenhouse in France, specifically for research. So out in the wild, it doesn't really happen. And so that certainly you get inbreeding through other ways, just not selfing. So you can totally have something as a parent, and that parent can also be a grandparent. That happens. In this case here, the techniques we used are they're kind of complex, but they're what we showed in that other slide. We look at the length of the shared fragments and whether there's evidence of recombination. And so in the case of yellow bellflower and Grimes Golden and their grandparent-grandchild relationships with the Rennet Franche, I looked very closely at the what's called the haplotype data to confirm that relationship. And at the same time, because you can have all sorts of crazy inbreeding in apple, there's a chance maybe the relationship is different, but it'd be unlikely to not be a grandparent-grandchild relationship.
Okay.
It also makes really good sense that yellow bellflower would be that close to Rennet Franche, because yellow bellflower, along with Tolman Sweet and Blue Pear Maine, 3 of the very oldest American cultivars. So having it be close to Rennet Franche historically makes really good sense.
Yeah, no, Grimes Golden itself too is quite old, if I recall, late 1700s. I think.
Yeah, I think so. Yeah.
Yeah. Yeah.
Okay, do you guys have more you want to present here, or should I take some more questions for you?
Yeah, give us questions. I think we've opened up enough cans of worms here.
All right. Well, first I want to throw in, I don't know if this is a question, more as a statement. Joseph Postman looked up Rennet Franche in the GRIN database, and the current accession there came as science from England, and they had a previous clonal introduction from France in 1937, but that no longer exists. And then Joseph did ask you guys, with late 20th century U.S. collections in Central Asia, particularly Kazakhstan, and theories about apples moving from Asia to Europe over spice routes, do you have any evidence of relationships between Kazakh apples and older European apples?
Yeah, so pretty much all apples that we eat are a combination of the Kazakh apple, the species is Malus sieversii, and then probably also the species Malus orientalis, which is from, it's in Turkey and the Caucasus Mountains, and then the European species Malus sylvestris. So those are the 3 main progenitors of Malus domestica, the apples that we eat. And so we can't really, that's the primordial ooze And a lot of that material wasn't preserved through grafting. So we can't tie it to exact individuals, but you can look at the genetics and find the evidence that they come from those, similarly to how in human genotyping services you can look at what your ancestral genetic makeup is, right? So I'm an American descendant from European ancestors, and I've got German and Polish ancestry in me, and they do that. The way that they do that is similar methods to how we can identify the distant makeup of apples. But when they're doing that in humans, they have way more SNPs, way better genetic tools available, better curated data, all this sort of thing. And
I'll ask you something about that, Nick. So in humans, it's usually large chunks, large haplotypes, large sections of the each chromosome that then add up together to provide those proportions of ethnicity or continental ancestry, etc. So for Apple, one thing you pointed out is the tool we use is a lot less resolution. But is it how much do you understand those chunks from originally from Seversii and from Sylvestris, a little bit of Orientalis, how much those have been chopped up into tiny bits, or are they still in large sections? So is it only half a dozen generations back, or are we talking several dozen generations?
I wouldn't be able to state the exact number or even a ballpark of the number of generations, but I can say that I've looked into this a little bit with the data that's available, and there still are some pretty large chunks. It's not completely fragmented, but then again, I'm just dividing the genome into whether you have or which species it is, and so you can have fragmentation between the same species. So you could have haplotypes of Malus Seversii kind of stitched together for a fragment, followed by Malus Sylvestris, the European apple, that sort of thing. I will say though that the older apples, particularly the really ancient apples, they do have larger fragments from Malus Seversii / Orientalis. With the tools we're working with right now, it's very difficult to distinguish those two species. We've developed a new array recently that might do a better job of it, that's what I can at least figure from the data right now.
Okay, so what that is indicating then is, at least in some cultivars, it is within a handful of generations, like half a dozen generations. But that's
the oldest material, sure, certainly. Yeah,
fascinating, huh?
Yeah, so I guess speaking of old apples, I see that there's a question here about Roman apples. So I was really interested in this topic, and so I made sure to genotype Dacio and Anurca, which are both apples that are recorded as Roman apples. Anurca it's interesting. You look at it, looks like an apple you'll pick right out of a grocery store, but supposedly it was recorded in Pliny, I think, and it's not at all related to any modern cultivars. It's genetically distinct, and but it seems a little different than Dacio. And Dacio is for those that don't know, it's recorded as the oldest extant cultivar, and it wasn't really used in breeding at all. It's not a very good cultivar. I actually have a few of the apples on my countertop right now because we have the apple in our orchard, and I want to figure out why this of all apples was kept around for such a long time, because it's not very good, at least the apples I've had. But I wanted to learn more about Italian apples, and I got a bunch of data on it, shared with collaborators, and we were able to do this imputation work to identify a common parent of a lot of old Italian cultivars, including Dacio. So we imputed a parent of Dacio, and there were so many offspring of this common unknown founder that I went over to visit some folks at the University in Bologna to try to learn more about the material. And through working with them, we did actually identify that individual, and we're working on a paper right now about the identity of it. So this would be the oldest cultivar, and its genetics are very, very interesting. It has very much it's much less derived from the European apple, the Malus sylvestris, as opposed to the Eastern progenitors.
So how long do we have to wait for that paper so you can get the reveal?
I have to wait for a Italian professor that sometimes is slow on responding to emails. So I have actually not even seen the manuscript.
Okay, so
yeah.
a while yet.
We identified the data. We identified the individual in using what's called SSR data. There's some SSR marker data sets, and we used a sort of like triangulation to figure it out. And then we found it out in the field, sampled it with a SNP data, and it was a match.
Great.
Yep, and I got pictures of it this year. It's a very interesting looking apple. Looks a lot like Dacio, except nicer.
Someone asked, how do you spell that apple that was mentioned in Pliny?
Annurca, Annurca. It's an excellent apple. It has an interesting story, so you have to check it out. I found it once in a specialty shop in Italy when I was visiting there.
Okay. So other questions will take us back a bit to some of the more previously mentioned things.
How much more time do we have? I forget.
Oh dear.
That's all right. We
done.
can actually go over a couple minutes. So we can probably go over about 10 minutes because we've got some great questions there. So if,
okay, well, I know that Cameron would like to speak a little bit about the how people interested in this could potentially be involved. And excellent. I see a couple of questions relating to that. So maybe Cameron wants to speak about that. I see one of the first questions here, Andrew.
Yeah, that was one I was going to get back to. Yeah.
Yeah.
So let me read Andrew's question, and then Cameron can tell us how we can get involved. Andrew says, I've got a number of seedling pippin apples that were found local to me that I've been cataloging for a few years. Most of them are likely 100-year or so old trees. A broad question is, what could a genome analysis service tell me about these apples other than the fact that they're not named varieties? Could such a service have any likelihood of telling me what named varieties these seedling apples are related to?
Okay, so yeah, sorry, can't help you. No, yeah, certainly can help. So this is what I was saying at the beginning, is we've opened this up. So it used to be only for the breeders and the researchers that we were doing this, and then we've now opened that up because that's right. There's all these trees out there, and you want to know, I want to know what they are. So let's figure it out. It's also helpful for feeding into the pedigree. Sometimes there are discoveries that are made from material that people have been sending in. So yeah, first of all, the identity, just checking if something matches something else directly and is another cultivar. That's really easy, and that could be done even 20 years ago with the SSR profiling panels. But now with the SNP arrays, we can very easily do parentage. Parentage jumps out really quickly and easily. Sometimes with really old ones, we're not sure which way round it goes, but as long as there are other related individuals, we can sort that out. Or if it's clear because of the historical information associated with one or the other, it's clear which is the parent, which is the child. And then we can also, well, pretty much in general, what we can do is find its pedigree position. So first of all, yeah, finding its parents. If we can't figure out its identity, just figuring out its parents already indicates, one or both parents indicates where in the pedigree it's sitting, and that gives a historical context to it. But even without parents, even if we can't find that, just about always we can figure out one of the, usually we can figure out one or more of the grandparents as well, and that helps place it in general. So some things really, most of the really old trees and seedling trees that people are sending in from the other side of the country there, the eastern side, since I'm in Washington here, Most of those eastern ones, old trees, tend to be like Rainet Frunch descended, grandchildren or great-grandchildren, so that shows where they are, really. But www.myfruittree.org gives the information about how to send in samples, and the fact that it unfortunately costs $120 per sample and can take up to 6 months for you to get your results. And I know there's some people here that are listening in right now who have stuff they've already sent in that are eagerly waiting the results.
Okay. Did you have more to say on how people can help besides that?
So John can say more, but I'll just say the old trees. That's the stuff. If you got old trees, especially if it looks like it's a cultivar, looks like it's grafted and being cultivated way back. Those are the great ones. That if it's not already in our data set and yet it's a cultivar, that's going to be very valuable. That's where it can most likely fill pedigree gaps, because the parents of things that the missing parents in most cases, I think, were actual cultivars. They're more likely to have been cultivars themselves. Go for it, John.
We've connected with people with apple collections in a number of locations around the U.S. and as Nick has mentioned, he's been working with people from all over the world. There are places in North America with collections that really should be part of this. One is the southeastern U.S., but there's probably other places as well. So those of you with large collections of apples might want to be in touch with us because some of what you have might be really interesting and useful in helping to put together this family tree of apples. That could also include, I noticed there was someone in the Q&A who mentioned Red Bay in Canada. It may be that if we're going to ever find any kind of connection with the Basques coming over, we're going to need to collect up there. So yes, we're looking for you if you have and you won't necessarily know if what you have is rare unless you're in touch with us. So you should definitely connect with us, and you can connect through NAFIX, and they can direct you to us. Also, I think for me, one of the most interesting things, and maybe Nick would say a little more about this, is finding the apples in England. For example, the other parent of Roxbury Russet, is it an Old English apple? And so one of the things that we need to do is to really explore some of the oldest European and English cultivars that we haven't found yet as well. So yes, getting involved with increasing the apples from more unusual areas would be a really great way for people to connect with us.
Yeah, and especially if you can find local funding, regional funding, etc., and you've got a collection, that's ideal. One-off trees, you can probably drum up the money yourself or whatever, but yeah, collections is that's going to provide the most amount of input on this. And so if you can find funding for that and get stuff then sent in, which dozens of groups have been doing that, then that's where we're making the most advances.
Okay, great. I have a question here that's harking back to the genetic guided breeding. Stephanie Blessing wanted to know if you can test the genetic markers in seeds. And I assume that's not true. You need the leaves. Is that correct?
I mean, there's DNA in seeds, but you wouldn't be able to get enough without destroying the seeds, I think.
Okay.
Technically, it would be possible, but not really with how we're doing it. Yeah. I mean, they get you can do DNA testings of babies in the embryo of humans, right? But we're not at that level with apples.
And then someone wanted to ask, why would present day Anna apples have no seeds?
I'm not familiar with
Anna. Yeah, I don't know
Anna,
it.
I guess. I know I have data for it. Yeah, I don't know. I'm not familiar enough with the cultivar to be able to answer that. I apologize.
So Anna tends to be really, really early season because it's low chill. And so also it tends to be early flowering too, right? So so one of the things probably that was done in, when it was a tree growing up in the family of other siblings that it was growing in, is it stood out as not only providing fruit early in the season, but overcoming some of the issues that other early ones had when they don't get enough pollination, for example. And so probably it can just pump out enough auxin itself to in its seeds from the existing seeds, or just internally in the core to develop fruit. So it's probably got something to do with being so early season and being a selected thing.
Okay. And then I think you answered this one, but Janet Federer says a 2011 German article is concerned with commercial apple inbreeding and says European apple ancestry is dominated by 6 varieties: Golden Delicious, Red Delicious, Macintosh. Jonathan Cox and James Grieve. Has subsequent work confirmed this?
Yes, I actually know the author of that. I think that's Hans-Joachim Bannier's paper, as a noted German pomologist. Pretty much, I'd say like every apple breeding program, every large apple breeding program uses the same base material. But every program has their own special sauce for the soup, So yeah, that's about right. Every single cultivar that you get in the market is descended in some way from Reinert Franch, pretty much. I don't think it's in the pedigree of Macintosh, so in the States that might be the one exception there. But every modern variety shares is connected to this like modern variety pedigree structure that includes those sorts of founders.
Okay.
Yeah, which is, required to make them, last, to be big enough, to be tasty enough, to last long enough, right? There's that base, which, as time goes by and as we dissect more and more, and the genetics underlying the basis of things that matter, we'll find hopefully smaller and smaller pieces that are required, so that they get locked in, but we don't have to lock in so much of the rest of it and have too much homogeneity there, that we can bring in lots of other parts to fill in all the other gaps. So there's already a lot of, as Nick said, source of other stuff that is used, knowing that we can have all this other diversity in the rest of the genome of apple. But yeah, the more we can limit down the parts that are required, the more we can mix up the rest of it.
Yeah, and even though a lot of those varieties are interrelated, there's still a lot of heterozygosity. So it's not like all modern varieties are all super inbred, and there's still a lot of diversity there. I would say that a lot of the varieties, though, aren't particularly robust, though, against challenges posed by climate change, and a lot of them aren't very ecological with regards to their resistance to disease. And sorry, I cut you off there, John. You had something to say?
Oh, that's okay. Well, I was going to say 2 things. One is a quick thing about how people could get involved, and that is that all of the identifications that are done through DNA profiles are done by matching, obviously. So one of the things that we're trying to do is to increase what we call the reference panel, or, which is the database to which your apple would get compared. And so one of the reasons, aside from understanding the sort of family of apples, for increasing, for submitting your apples, is if they are not ones that we've encountered we as historians and as geneticists are working together to increase the database to which to compare. That's another reason to get involved. And then I think the breeding question is sort of a good segue into at least a few comments about Honeycrisp and how Honeycrisp is somewhat different, and maybe Nick or Cameron, you could speak to that, is somewhat different than some of these others that were mentioned, and how is it now playing a huge role in the breeding of the apples of the future?
Or maybe I'll say something there. Nick might have his hands tied on that one. So yeah, so it was serendipitously Honeycrisp. So Honeycrisp has got this ultra crisp phenotype to it, fruit quality, which is really amazing. It provides crispness without firmness. That's otherwise what had been going on for the previous couple hundred years. To get crisper and crisper apples, everybody loves crispness. They were getting firmer and firmer until there's in some breeding programs, they just know they get material that's just way too firm. That's rock hard. Even though it's very crisp, it's rock hard, and they know, okay, we're not going to put that out as a new cultivar. So Honeycrisp has this. It was the first time to introduce this great crispness while not being too firm. So universally awesome. People really like that. And it turns out, as we looked into the genomic background of it, that looks like it comes from a couple of genomic segments that have come from a wild crabapple. Ultimately, and it wasn't intentional. It would just happen to be there and mixing up in the background of old Minnesota material 100 years ago. And so, yeah, an example of the value of bringing in really, really diverse stuff there.
I have to ask, because I looked at that paper on the Honeycrisp family tree, and I see no crabapple in that family tree.
No, because in that paper, we only go back. Nick only took it back just to show back couple of generations, right? Because I was just trying to fill in that missing father was the main thing about that paper. But it's via frostbite. So frostbite is it's not a crab, but it's partial crab, so basically. So it would be a great grandparent or great great grandparent, I think, of frostbite is the estimate, because I think frostbite has 6%, something like that, 6% crab in it.
I see.
And yeah, so great great grandparent is probably what there is. So several generations.
Well, I'm sorry we don't get to all the questions, but we are going to have to call it. We're quite over time, and we do have another session coming up soon, our keynote speaker sessions. We got to get people able to attend that. So on behalf of NAFEX, I want to thank you all for making this a great session. Once again, this session is being recorded. It'll be made available on nafex.org webpage in a few days, and then in a year it'll be on YouTube. There may be other downloadable content on the webpage along with this session that you can access to find out more. Don't miss out on our next session, and I hope you really enjoyed our conference, and please stay connected to us on social media and other places. And thank you all for joining us.
Thank you very much.
Yeah,
Thank
thanks
you,
everybody.
guys.
Yeah, thanks.